Hi everyone. So in this lecture, we are going to be looking at light in relation to plant growth and development. And this is one of two lectures in which we'll be looking at light. But first, we will review. So in last lecture, we started talking about plant growth and development. And we said that plant growth is an increase in plant size due to cell division or enlargement, whereas plant development is the progress of a plant through its life cycle, from seed to seedling to young plant to mature plant, etc. Now, we then started by looking at the genetic factors that affect plant growth and development. And we started by defining what a gene is. And we said that a gene is simply a segment of DNA that codes for a protein. And it's that protein and the interaction with the environment that creates a trait. Now, DNA is our hereditary material, deoxyribonucleic acid, and it consists of two sugar phosphate backbones that are joined together by a series of nucleic acid bases: adenine, thymine, guanine, and cytosine. Adenine and thymine always join together, guanine and cytosine, always join together. And it's the series of these bases, right, the particular code that actually codes for the particular protein that interacts with the environment to create the particular trait, right, at its most basic. It gets more complex, but that's the most basic version of what is going on. And if we take the entire genetic makeup of an individual, all of its genes, we will say that is its genotype, right? Or its blueprint, you can think about it, the genetic blueprint. Whereas the phenotype is the actual observed traits, right? And that is based on the genotype plus the environment. And we said the environment can interact or can affect the phenotype in a number of ways, right? So you can have the same genotype, but due to different environmental conditions, you will see different observed traits. And we refer to that as phenotypic plasticity, right? Which is a change in the physiology, morphology, or behavior of an individual in response to different environmental conditions. And we looked at a couple different examples. We saw watercress, and we said that the temperature at which it's grown will affect the leaf shape, right? Whereas you can get either entire leaves or very divided leaves based on temperature. We saw the effect that light has on the height of seedlings, right? With an increase in light, seeing a shorter and shorter seedlings, right? Or a decrease in light, you see taller seedlings, right? Examples of this plasticity and phenotype. And we gave a couple other examples as well. And the last thing we looked at were transcription factors. And this is really, really important because this is one of the ways, or transcription factors are one of the ways that we see this phenotypic plasticity, right? It's one of the underlying mechanisms behind this. And what transcription factors are special proteins that turn genes on or off. So what is occurring with some of this plasticity or some of these environmental effects is that there is some environmental effect that is being picked up by the plant that triggers these transcription factors to either turn certain genes on or turn other genes off. And that is going to result in different traits or different morphological responses, different physiological responses, or different growth patterns, right? In terms of whether our plant is taller or shorter, or how its leaves look, etc. Right? So that transcription factor, very, very important. Okay, now on to the lecture. So I want to start by looking at what light is. So light is a type of electromagnetic radiation, which is essentially energy that is propagated through space in waves. And it's the length of the waves, or the distance from crest to crest from each wave, which we refer to as wavelength, that is going to determine the type of energy. And if you look on that image that I have up there, you could see on the one end where there's very, very short wavelengths, right? So the distance between waves is very short. You have gamma rays, and it gets a little longer to X-rays, ultraviolet, ultimately visible light, etc. And as your wavelengths increase, you could see some other types of radiation, including wireless, TV, radio, MRI, etc. Now, if we are talking about the energy that is produced by the sun, our sun is producing about 2% UV light, right? 47% visible light, and 51% infrared light. We refer to this as the solar spectrum. And if we focus on that section of visible light, we can see that again, our wavelength is going to influence radiation. This time, it's not influencing the type, right? This is going to be all classified as visible light, but it is going to influence the color of light that we see, ranging from purple or blue on one end, on the shorter end length, to red on the longer wavelength end, right? And really going from around 400 to 700 nanometers. Now, light is critical for plant growth and development. And Dr. Bruce Bugbee from Utah State University has developed these 9 cardinal parameters that are required or greatly influence our plant growth and development. And you can see that light is listed there at the top. And the reason why light is so important is because plants use light energy to create glucose or sugar in a process called photosynthesis. And in this process, our plants are taking carbon dioxide from the air and they are fixing it. So they are taking it from this gaseous form and they are making it into a solid, and that solid molecule is a glucose molecule, right? So they're using carbon dioxide and water in this process and energy from light, and we refer to that as photons, little packets of light energy. And they're creating that glucose molecule, C6H12O6, and producing oxygen as a byproduct. And this is really important because ultimately, photosynthesis is the primary factor that determines the yield and quality of plants and their products, right? And it's photosynthesis that is going to influence plant growth and plant development. And glucose is so important, and it's because this glucose is so important, right? Glucose is stored as food. It is broken down into energy. So if your plants need energy for various metabolic processes or pumping in nutrients from the soil into their root system, they require energy, and they're going to need glucose or the breakdown of this glucose to get that energy, right? And that glucose is also turned into a number of other compounds through these various metabolic processes that also require energy, right? So in order for your plant to grow and develop, it's going to need energy. It gets that energy through the breakdown of glucose, and that glucose is produced through this process of photosynthesis. And in order for that process of photosynthesis to occur, we need light. But are all wavelengths of light equal for photosynthesis? Right? We just saw how there are different wavelengths of light that determine, at least when we're talking about light, the color of our light. Are all those colors the same for photosynthesis? Well, early experiments that were done in the 1970s showed that chlorophyll absorbed a large percentage of red and blue light and little green or yellow light. And chlorophyll is the pigment that is found in the chloroplast, which is the organelle in the cell where photosynthesis is taking place. And when they did those experiments, they saw that this chlorophyll was absorbing this red and blue light and little green or yellow light. And this gave rise to the idea that plants only use red and blue light for photosynthesis. However, when they started doing plant growth experiments using just red and blue light, or red and blue LEDs in this case, they had poor results in plant growth. And the question was why? Well, they were only utilizing, in those early experiments, they were only utilizing chlorophyll, and they were liquefying the leaves in this process. But when they did a similar experiment utilizing the whole leaf, they saw different results, right? And they saw that there was some absorption of green and yellow light when they were utilizing the whole leaf. And if you look at this graph up here where we see chlorophyll absorption, and then we see photosynthesis rate as a function of absorbed wavelength, you do see that photosynthesis is occurring at these green and yellow wavelengths, right? Maybe not as high at red and blue over here, red over here, but it is occurring, right? So what was happening here with this whole leaf? Well, one of the big reasons why we see this difference, it has to do with the ability of green light to transmit into deeper plant tissues. right? Red and blue light, because it is absorbed so quickly by our chlorophyll, gets absorbed very fast, and it gets absorbed at the surface tissues, or at the tissues that are closest to the surface of our plants, right? So these cells, if we're looking at our leaf right here, it's going to get absorbed high up here. Green light is able to transmit deeper into the plant tissue. So it's able to get into cells or deeper cells, and those cells are able to photosynthesize that they otherwise wouldn't be able to if they were just if we were only growing with blue or red light, right? Because it's going to get absorbed very high up or at the surface of those tissues. And this is also true for maybe leaves that are covered, that are lower in the canopy, right? So those leaves up on the top part of the canopy are absorbing the red and blue light preferentially. Little of that is getting down to the lower leaves or stem tissue, and thus if you're just using red or blue light, you're not maximizing your photosynthesis. If you include some green light, some of that can transmit into deeper plant tissues or deeper plant cells and into structures that are below other structures or below canopy structures. So improving the rate of photosynthesis. And this gave rise to this idea of photosynthetic active radiation. So these are wavelengths of light that are important for photosynthesis. And you can see it's going to range from around 400 nanometers to 700 nanometers, right? These are the wavelengths of light that are critical for photosynthesis. We refer to this as photosynthetic active radiation. So the next question is, how much light do our plants need? And we can refer to this as light quantity, right? The measure of how much light is produced, right? So we have different ways of measuring light quantity. Now, originally, we based all that measurements on the lumen, right? And the lumen is a measure of light output that's visible to the human eye, and it's related to the amount of light that is produced by a single candle, right? So if you have a single candle, the amount of light that we see produced by that single candle is considered 1 lumen. And then based on this concept of lumens, we developed some other measures of light intensity, foot candles and lux. So foot candle is the amount of lumens or the intensity of light if you have one candle or one lumen that is set a foot away from a wall. And if you're viewing the light intensity or the amount of light on a 1 foot by 1 foot section, this would be considered a foot candle, right? So you have your candle, it's a foot away from your wall, you're viewing an area that's a foot by a foot, and the intensity of light that you see there would be considered a foot candle. Lux is essentially the same thing, but in kind of the metric form, right? So in this lux example, your candle is a meter away from the wall, and you're looking at a meter by a meter foot square area, and the light intensity that you see on that area would be considered 1 lux. Now, the problem with lumens, and therefore foot candles and lux in regards to horticulture or agriculture or growing plants, is that those measurements are based on the human perception of light instead of what the plant sees or instead of what's important for the plant, right? We said that what is important for the plant in terms of photosynthesis is that photosynthetic active radiation, the range of light from 400 to 700 nanometers. And the problem with lumens is that our visual perception does much better at being able to see within the green and yellow light spectrum. So right here, if you see lumens right here, this is what we see when we are utilizing our visual perception. So we don't do as well at being able to pick up the amount of blue or red light. And that is a problem, right? Particularly because we just talked about how blue and red light is preferentially absorbed by our chlorophyll, right? It is utilized by our plants for photosynthesis. So if we are basing our measurements on lumens, we may have a high lumen content, but that may just be a high green or yellow content that maybe isn't ideal for our plant growth in terms of our plant being able to utilize light for photosynthesis. Instead, we developed other measurements that are based on that PAR value, that photosynthetic active radiation value. And they are photosynthetic photon flux and photosynthetic photon flux density. So photosynthetic photon flux, or PPF, is just a measure of the total amount of PAR, so the measure of the total amount of light within that PAR range from 400 to 700 nanometers that is being produced each second by a light source, right? Whether it is the sun or a grow light or a lamp, etc., right? And that is expressed in micromoles per second, right? So the number of micromoles per second of light in that range, that PAR range, that is being produced. Photosynthetic photon flux density is the amount of light in that PAR range in micromoles per second that's being produced, that's arriving on a square meter area, right, every second, right? So that photosynthetic photon flux density is a very similar measurement to foot candle or lux, but instead of being based, really it's more similar to lux, right, because it's based on that 1 meter by 1 meter area. But instead of being based on lumens, it is based on PAR, which is that range that's important for your plants for you to utilize photosynthesis. So given that, right, and we know that photosynthetic photon flux density is the better measurement if we're going to measure the amount of light or the light quantity that's being produced by a lighting system. Given that, how much light do our plants actually need? Well, it's going to vary based on the species of plant, right? But often, as a generality, there's going to be a curve that is similar to what you see there on the left, right? That as you increase your ppfd values, you're going to see an increase in photosynthesis and thus an increase in growth or ultimately yield. But at a certain point, it's going to level off, right, for most plants. And it seems that as a generality, that level off point is around 1,000 ppfd. For reference, the sun produces on a sunny day 2,000 ppfd, right? That would be the measurement on a sunny day. So if you get around 1,000, that is going to maximize, for most plants, your plant growth, right? Or at least your photosynthesis. That thus anything that's affecting photosynthesis is going to affect plant growth. But like I said, there are variations based on species, right? Some species require less light, such as lettuce, and you can see that as you increase with lettuce, really it's around 500 you're going to maximize. And if you start to get too much, you can see a decrease in photosynthesis. But other species respond much better to increases in light or light quantity. And cannabis is an example, or corn, right? Where they can take in a lot of light and really, you can really, by supplying them with a lot of light, you can really maximize their growth. Now, ppfd is a really important value, right? And it gives us a lot of information, but it doesn't tell us, it only measures per second, right? It's the amount of light that the plant is getting each second. But really what's more important is over the course of a day, how much light our plant is getting, right? Because if our plants just got 1,000 ppfd for about 5 seconds for the entire course of the day, they are not going to grow and develop very well, right? So we really need a measurement that takes into account the amount of light that your plant is getting each day. And that measurement is referred to as the daily light integral. So like I said, this is going to measure our ppfd over the course of the day. And it's calculated relatively easily. You can see the full calculation or the full equation up there. But in practice, all you need to do is get your ppfd value, multiply that by the times of hours of light that your plants are exposed to, and then you multiply that by this constant of 0.0036. And that's just a way of changing seconds into hours, essentially. But if you do all that, you will get a value, and that is your daily light integral value. And what is helpful is that there's information available that looks at different plant species and tells you what you need in terms of an average daily light integral value to produce quality products. Now, here's an example of that chart, and you can see if you went to this link here, you would get more information. But in general, you see above 14, you will have good quality. So you want that as a minimum of your daily light integral, and above 22 is even better. Now, if we were to grow outside, we are going to see that our daily light integral is going to vary by season, right? And this has to do with the angle of the Earth or the Earth's tilt and how that relates to seasons, right? So during the summer, where you have hotter temperatures and where the sun is more concentrated, leading to those hotter temperatures, we're going to have a higher daily light integral, right? And that is reflected, especially where we are. If you look on these maps, right, you can see that as you go from winter into spring and summer, your daily light integral increases correspondingly. And to go back to, if we were talking about maximizing growth, right, for most plants, as an example, if we were growing at our 1000 ppfd, which we said is kind of like a good standard to go for, you can see how your daily light integral is going to change by the amount of time that in this case, that your light is on, or maybe the length of day, right? So at 16 hours a day, if you do your calculations, you're going to get a daily light integral of 57.6, which is very, very high, right? At 12 hours a day, that drops down to about 43.2, right? So you can see how it's not only the ppfd value that's important, but it's also the amount of time, or the length of time, that your plants are exposed to that light. So that brings us to the last part of this lecture, which is then how do different colors or wavelengths of light affect plant growth, right? We know that these entire wavelengths are important for photosynthesis. They're all utilized in that PAR range. Well, are there specific effects of specific colors of light? And what we see is that light quality, which is, we refer to light quantity as really the intensity of that light, right? The ppf or ppfd values. Light quality is the spectral range, right? So what wavelengths are included. So light quality or wavelengths have a bigger effect on plant shape than they do on plant growth, right? They're not really going to affect your plant growth, but they may affect the shape of your plant. We're going to look at some different colors. And you see, if we are talking about blue light, the main effect of blue light is that it creates shorter plants. And you can see that here, where these plants were grown under different percentages of blue light. And you can see the relative sizes, right? From 5% blue light to 20, you see a much shorter plant. And here again, as we increase our blue light, we see correspondingly smaller plants, right? Now this is affecting the shape, right? But it's ultimately not really going to affect your yield in terms of plant mass or product, right? It's affecting shape rather than growth in terms of yield or productivity, right? And we can see that with these examples here that were done on cannabis, right? So here were different percentages of blue light for cannabis plants were grown under different percentages of blue light. And you can see that the yield, really, in terms of the flower yield, did not vary significantly between them, right? There is some variation, but that's natural variation that we would expect. And there was no significant or statistically significant difference between yield amongst these different treatments of blue light. And that goes to the same not only for yield of flowers, but also cannabinoid concentration as well, like CBD and THC. There was no statistically significant difference in those concentrations with plants that were grown under these different concentrations of blue light. Right, so it affects shape, not so much yield or productivity. Green light. Green light, again, is utilized for photosynthesis, right, to an extent, right? It's able to transmit through leaves. But green light is really important in terms of being utilized for growth, or in this would be growing indoors, right? Type of system. You want to include green light because green light helps you identify plant problems and diseases, right? And if you can see, one of the problems with growing just under red light or just under blue light or even just under white light is that it makes it hard to identify potential diseases or potential deficiencies in your plants, right? Those are often observed. We often see those deficiencies as changes in color of our plant leaves. And if you're growing without green light, you're not really going to be able to see those changes. Thus, it's important to include in a lighting system if you are growing indoors. Red light. Red light does have an effect on photosynthesis, and thus a slight effect on improving yield or growth. It's about 15% better for photosynthesis, or more efficient for photosynthesis, than blue light, right? So including red light, you do get a little bump in growth, not an extreme amount, but it does help. And then far red light, which I haven't really talked about yet, but far red light is beyond that 700 nanometers, so something like maybe 740 or 750. That, including that, will make your plants taller. And that is a function of the shade avoidance response, or photomorphogenesis, which we'll learn about in the next lecture. But if you include some far red light, it is going to make your plants a little bit taller, right? And you can see that In these experiments up here, look, as you increase your amount of far red light here, FR, you see that your plants correspondingly get taller, right? Red light also has an interesting effect called the Emerson effect, where you can actually increase the amount of photosynthesis when you add a little bit of far red light, right? Um, and it's because of this Emerson effect, right, that we see this, and it's related to photosynthesis. When we get to that later on in this year, I will touch on this again of the mechanism behind this. But adding that far red light gives you a boost in photosynthesis as well, and it's because of this now extended this PAR range to include far red light. So traditionally that PAR was from 400 to 700 nanometers, and that's what we considered important for photosynthesis. We now realize that we need to extend that out to around 750 nanometers to include that far red spectrum, and this is referred to as EPAR. And if you look at a lot of lights that you could buy nowadays or lighting systems, they will say that they're including infrared light or far red light because of this EPAR and because of this effect. Now, if we go to the opposite end of that, right, the far red light has the longer wavelengths. The shorter wavelengths beyond visible light would be your UV light, right, on the other end of that visible light spectrum. What effect does UV light have on your plant growth? Well, this is really the cutting edge of science right now in terms of plant science and looking at light and how it affects our plant growth. And what we see is there are different types of UV light, A, B, and C, with B and C being most damaging. And they have found that short exposure of plants to UV light does seem to be beneficial. It does seem to boost plant growth or plant yield. However, you have to be very, very careful because this is very, very short exposure. It's minutes to seconds with some of the more damaging B or C. I think it's seconds even, right? And I think UVA may be minutes, but it's very, very short exposure. Beyond that, you can get too much damage to your plant. It can damage your plant cells and tissues. So short exposure of UV light does seem to provide some benefit in terms of your plant growth. However, again, this is relatively new and cutting edge, and we don't have all of that worked out quite just yet. So that is it for light. I will see you guys on the next lecture.